Good morning, class. Chapter 7, Part 2. We're going to pick up with osmotic pressure. Osmophiles live in habitats with high solute concentration. They are obligate halophiles and facultative halophiles. Who can tell me the difference in the word obligate and facultative? Obligate requires high concentration of salt for growth. It has to have 9 to 25% sodium chloride. It has significant modifications to their cell walls and membranes and will lyse, break, in hypertonic habitats. Facultative halophiles are resistant to salt, even though they do not normally reside in high salt environments. Radiation, phototrophs can use visible light rays as an energy source. Some protective measures against radiation. Yellow carotenoid pigments absorb and dismantle toxic oxygen. Other microbes use enzymes to overcome the damaging effects of UV radiation on DNA. Just from Biology 103, what's an example of a yellow carotenoid? Look at the word, C-A-R-O-T. Yes. Hydrostatic pressure. Barophiles live in deep sea microbes that exist in pressures up to 1,000 x atmospheric pressure. So where do they live? In the deep sea. So strictly adapted to high pressures that they rupture when exposed to normal atmospheric pressure. Barophiles, deep sea microbes that exist in high pressure. Pressures up to 1,000 times the atmospheric pressure. Some associations between organisms. They can either be symbiotic, which means they live in close relationships and nutritional relationships, and it's required by one or both members, or non-symbiotic relationships, which organisms are free living, not required for survival. Who's my other class that starts on 62? This class was supposed to start on 72, so this is a review. So in symbiosis, who benefits in mutualism? Both members. In commensalism, who benefits? All right, and what about the other one? They're not harmed. And a parasite is dependent and it benefits. But what about the host? The host is harmed. Two types of non-symbiotic relationships are synergism and antagonism. Antagonism, against. Some members are inhibited or destroyed by others. What could they be competing for? They could be space, nutrients, nutrition. Biofilms are the epitome of synergy. They are mixed communities of different kinds of bacteria and other microbes. Here's an example of one. Pathogenic microbes have an optimum growth temperature in the range of what to what? Degrees Celsius. Say it again. What kind of anaerobe can grow in the presence of oxygen but does not require it? The difference in the words what? Obligate and facultative. Obligate is kind of like the word obligation. Must have. If it is an obligate halophile, it must live in a salt environment. Facultative it's a wider niche. I don't have to have it, but I can grow in the presence of it. So what is number 2? What kind of anaerobe can grow in the presence of oxygen but does not require it? Facultative. Number 3, obligate what? Inhabit. Barophiles. In a what kind of relationship one member benefits and the other is harmed in some way? Yes. Here are our learning outcomes for the study of microbial growth. We're going to summarize the steps of cell division used by most bacteria, name another method used by fewer bacterial species, define doubling time. At the end of chapter 7, there is a question about under the critical thinking, because I want you to become critical thinkers, not just memorizing material, that deals with doubling time. And it's on the quiz. I just don't have it to read it to you right now. So when we get to it in this section 7.3, we're going to take the time to really understand it. It's all about this girl that brought some pasta salad to the class picnic. And she left it out on the table for 3 hours. She introduced 20 microbes into it. And so the question has to do with, what is the total number of microbes at the end of 3 hours? So just keep that in mind when we get to it. We're going to compare and contrast the 4 phases of growth in a bacterial growth curve, identify one quantitative and one qualitative method used for analyzing bacterial growth. Well, the basis of population growth is binary fission. The parent cell enlarges, the chromosomes are duplicated, the cell envelope pulls together in the center of the cell to form a septum. See this protein band right here in the center of the cell? And in the very next step 4, that septum right there forms. Can you see what I'm pointing to? Okay. The cell divides into 2 daughter cells after it forms the septum. Here we go. Generation time or doubling time. So doubling time means the time it takes for a totally new generation to occur after binary fission. The process. It's the time required for a complete fission cycle. Each new fission cycle doubles the population. As long as the environment remains favorable, the doubling time can continue at a constant rate. The length of the generation time is a measure of growth rate of an organism. Exponential growth. What's another word for exponential or exponents in math? Say it. Yeah, it expands, but how does it? It expands exponentially, or another word I could say is logarithmically. Logs and exponents mean the same thing. So the exponential growth, the growth pattern of microbes, is useful to express the population of microbes as exponents or what? Logarithms. Plotting data from a growing bacterial population, the number of cells as a function of time, and it can be represented logarithmically or arithmetically. The population growth curve, a predictable pattern of growth in a population. There are different kinds of counts that are used to figure out how many are in this population. The viable count technique is traditionally used to observe the population growth pattern. The total number of cells is counted over a given time period, and there's a fundamental method of laboratory microbiology. So here are the steps in a viable plate count method, the batch culture method. Look up top, we have the time. So time is important here. 60 minutes, 120 minutes, that's 2 hours, 3 hours, 4 hours, 5 hours, all the way out to 600 minutes, which is how many hours? Thank you. All right, and here some plates are incubated. Let me move this. And the colonies are counted. At first, there's none. Then you see these 2 little colonies right here, And here there are 4 colonies, and they continue to grow how? Expo who? And this is the total estimated population in a flask. So this is the growth curve. First we have a lag, not a log, but a what? A lag. Now what happens during the lag phase? Take your finger with me so that you understand exactly what this is doing, because I'm going to ask you in just a minute. Lag phase is just kind of going straight across at a flat rate. And then what's the next phase? Exponential growth. Go way up. Exponential growth. It goes up high. And then what happens? Stationary. What happens during the stationary phase? Is it growing? Okay. And then what happens? Death phase. And I'm going down. All right? But now, are all of the microbes going to die in the death phase? Does it look like it? Okay. What are the stages? Lag, exponential growth, stationary, death. Lag is flat period on the graph when the population appears not to be growing. Newly inoculated cells require a period of adjustment, enlargement, and synthesis. Cells are not yet multiplying at their maximum rate. The population of cells is so sparse that the sampling misses them. Exponential or log phase. It's the period during which the growth curve increases geometrically. The phase will continue as long as cells have adequate nutrients and the environment is favorable. When you do your lab today, you're going to inoculate your Petri plates with bacteria from all natural sources. The air in the room, the air outside the room, the air outdoors, several others. But when you incubate it and we take it out Thursday, if you have so many colonies that have grown that is not any more space on there, what would be a problem with why they couldn't anymore grow? They were competing for space, not enough space. All right. The phase will continue as long as the cells have adequate nutrients and the environment is favorable. Then the stationary growth phase, the population enters a survival mode. Cells stop growing or grow slowly in death. Limiting factors intensify and cells begin to die at an exponential rate. The curve dips downward. So the practical importance of the growth curve: antimicrobial agents rapidly accelerate the death phase. Microbes in the exponential phase are more vulnerable to these agents than those in the stationary phase. Actively growing cells are more vulnerable to conditions that disrupt cell metabolism and binary fission. Growth patterns can correspond with the stages of an infection. Bacterial shedding during the early and middle stages of infection is more likely to spread it to others. Chemostat is another system, continuous culture system that emits a steady stream of nutrients. It siphons off used media in old bacterial cells to stabilize growth and cell number. It maintains the culture in a biochemically active state and prevents it from entering the death phase. Other methods of analyzing population growth. Today, when you use a swab, a cotton swab on the end of a stick, and put it down in a liquid growth medium in a culture tube, you're going to express that extra liquid off of it by pressing it up against the side. And let's say, and it's a clear, it's kind of brownish, but it is nothing in it. It's a clear liquid, clear brown looking liquid, and it's nothing but liquid agar, TSA, tryptase soy agar. But after I have expressed it there, gotten all the extra liquid off, and put it behind my ear, what do you think I'm going to get? Rub it down like that. Then put it back in there. Now, if the stick is too tall to close for the top to go on it, what are you going to do? Break the stick. You can just take it with your fingers and break the stick. Close it up. When we put it in the incubator, by Thursday, 48 hours later at 37 degrees Celsius, we take it out and look at it, it will be turbid. What does that mean? If I look at it and it's turbid, it's cloudy. And the more cloudy that it is, the more bacteria that grew. Okay, because of whatever I got off my ear. Now, some people in my other class didn't want to put it behind their ear. The book suggests that you can use a doorknob or a phone. I just think it's interesting to see what grows from behind your ear. But what are we going to do? We're just going to follow the book. So turbidometry, all of that to say, a tube of clear nutrient solution becomes cloudy or turbid as microbes grow in it. That's how you will tell, did any microbes grow. The greater the turbidity, the what? The larger the population size. Then there's direct total cell count. Cells in a sample are counted microscopically. It utilizes a cytometer, calibrated to accept a tiny sample spread over a pre-measured grid. Here is a picture here of turbidity measurements as indicators of growth. Here we have low turbidity, but look how cloudy this one is. High turbidity, lots of growth. Here's a direct microscopic count of bacteria, and you are going to count some colonies to tell me, and I'm going to put it up on the board, how many colonies each of you grew. And that's going to become our data for our results. All right? So there are many different types of methods to count these, but we're just going to use the old eyeball on it. You can count that many right there, can't you? Now, if it was thousands or if it was hundreds, it might be difficult to count. But you're just going to count them by looking at them and counting. The old-fashioned way. A culture counter electronically scans a culture as it passes through a tiny pipette. Look here at all these little microbes that are going through that pipette down into this electronic detector. Right there is where it's going to count them. Each cell is detected and registered on an electronic sensor. A flow cytometer is similar to a culture counter. It can measure cell size and differentiate between live and dead cells. That's a clue there now. A flow cytometer does basically the same thing as a culture counter, but it can differentiate between live and dead cells. So when I ask you that again in about 2 minutes, you'll be able to tell. So some new methods of analyzing population growth include polymerase chain reaction, quantifies bacteria and other microorganisms in environmental and tissue samples without isolating and culturing them, and tests that measure ATP are used in food and pharmaceutical industries, may be used for rapid quantification of microbes and other environmental samples. The average generation time of most bacteria under optimum conditions is 24 to 48 hours. True, false? Everybody together now. Place the 4 steps of the growth curve in the proper order. Okay, ACDB. Let's just say it out in words. The lag phase. What can differentiate between live and dead cells? A flow cytometer. What about a culture counter? Can a culture counter do that? That's exactly right. Thank you, ma'am, for listening. And that's it for chapter 7. I'm trying to think now here, how do I just pause?